Hello students. We all know that an apple a day keeps the doctor away. This sentence is very much true when the apple is available to us throughout the year. But is it really possible? Let us learn about the different techniques by which we can really preserve our foodstuffs for a long period of time in order to increase its shelf life. Today we are going to deal with the different physical methods by which we can preserve the food. We consume variety of food and variety of beverages in our daily food item. Now this food is associated with another biological entity, and that is microorganisms. If the microorganisms are growing inside this food, it is not necessary that it will spoil the food. Rather, it can also make the food better if it is a fermented product. But if it is not a fermented product, the undesirable or pathogenic microorganisms grow inside the food and spoil the food, and therefore preservation holds a very important place in the storage of the food items. Let us first learn about the significance of the preservation of the food. Now, each and every food item is not available throughout the year. Therefore, when the production of a particular food takes place in a massive amount, we should be able to store it so that we are able to consume it at that time when it is not produced or harvested. For example, the potatoes are produced in a mass amount during winter months. Now, are we able to consume the whole potato that is coming into the market, but rather we try to preserve it? So, the first significance of preservation is we want to take care of the excess food that is produced at a certain period of time. The second or most important significance of preservation is it adds variety to our meals. Could you think of a day when you are going to have the same food beginning from your breakfast till your last supper? No, it will not be desirable for you because we want to have variety in our meals, and that is only possible when we are able to preserve our food for a much longer time. Also, preservation reaches the food to so many areas where that particular food item is not grown or not produced. For example, if I take a tropical fruit, mango. Now this mango or banana is typically a fruit of tropical countries, which is not available to the people of temperate region. But if preservation is done in a proper way, it could be made available to such areas where it is not grown. And the last most important significance of preservation of the food item is it makes the transportation and the storage of the food items much easier. If you are going to store kgs and kgs of potato, it becomes a massive amount. But if you are able to preserve the potato in the form of flakes or powder, the transportation as well as the storage of potato becomes easier. Before going further, we say that each and every food item has got a shelf life. Now, what is this shelf life? Shelf life means you are going to retain the aroma, the taste, the color as well as the nutritive value of the food time for a much longer time so that the food becomes safe for the consumption to the human population. According to the shelf life of the food, we divide the food, or rather we classify the food items into three major categories. They are as follows. Number one is the category of non-perishable food. Non-perishable food means the food which is not so easily destroyed or degraded. You are very well aware that in our homes we store potatoes, onions, garlic, ginger, rice or many dals in normal room temperature. We do not store them at refrigerator nor do we froze them. That means such food items which have got a longer shelf life at a room temperature without any particular conditions of preservation are known as non-perishable food items. Going on to the second category of food, which is called the semi-perishable food. Now, semi-perishable food means these food items have got a little amount of moisture along with the fat content or lipid. Now, since they have a fat or a lipid in their constituent, they are not degraded or are not destroyed so easily. So, they do not need refrigeration immediately. But they cannot be stored too long if they are opened and consumed. So, once they are exposed to the air or to the environment, they need to be preserved, and therefore they are known as semi-perishable food items. For example, your cheese and butter. Going on to third category, that is called the perishable food items. Now, these food items are not at all stable in the room temperature. For example, you could recollect that if you have fresh meat or fish or fresh vegetables and fruits which cannot be stored at room temperature, they need to be refrigerated at a lower temperature at about 4.4 degrees Celsius, or sometimes the meat and fish has to be frozen when they are kept in the refrigerator at -20 or at -15 degrees Celsius. That means such food items are not at all stable in their room temperature and they do not have a shelf life. Therefore, such food items are called perishable food items. Now, according to the different groups of the food items that is available to us, we have different methods of preservation. Why do we need the preservation technique? What exactly is happening inside the food that needs to be preserved? Now, let us learn the biological basis of preservation. Now, when I talk about food, the food is associated with the humans because humans are not able to live without food. In the similar way, the food is associated with the microorganisms because the food is a very good source of nutrition for the microbes to grow and to survive. Therefore, the microbes always want their population to increase, and this spoils the food. So, when we talk about the biological basis of preservation, we have to take care of two important things. Number one, the microorganisms which is present inside the food and the certain enzymes that is present inside the food which may degrade the food. That is called the self-decomposition of the food. Now, let us learn about these two factors a little detail. In biological basis, we say that we want to maintain the asepsis. That means the septic condition should be maintained. We should keep out the microbes. Either we could inhibit the growth of the microbes, that is, we say that we inhibit the microbial growth, that is referred to as microbicystatic, or in other way, we want to destroy the irreversible inactivation which is called the microbicidal. So, either we restrict the growth of the microbe or we kill the microbe. Another aspect or another factor that we take into account is we want to prevent the self-decomposition of the food. Now, this self-decomposition of the food is taking place because of certain enzymes which is present inside the food. Now, these enzymes have to be inactivated. So, we say inactivation of the food enzymes as well as the prevention of oxidation by inserting certain antioxidants or eliminating the pro-oxidants which may increase the oxidation process. Now, whatever we want to do, our basic purpose is combining different methods together so that we could inhibit the growth of microbe. On the other hand, we could inhibit the activation of the enzymes, and these two together therefore help us to preserve the food and to reduce the moisture content and to increase the shelf life of the food item, whether it's a perishable, non-perishable or a semi-perishable food. Let us understand the principles of the food preservation. Now, certain principles have to be followed. Number one is prevention or delay of the microbial decomposition. Now this prevention or the delay of the microbial decomposition can be done by various methods. We can do it by keeping out the microorganisms, which is called asepsis. This can also be done by removal of the microorganisms by means of a technique which is called filtration. We can also hinder the growth and the activity of the microorganisms by lowering the temperature, by process of drying, or by maintaining the anaerobic condition so that the oxygen supply is not given to the microbe and the microbe will not grow. Also, we can kill the microorganism that is present inside the food by means of heat or radiation. Second most important principle of preservation is prevention or delay of self-decomposition of the food. That means this delay of the self-decomposition can be done by destruction or inactivation of the food enzymes by blanching. Now, blanching is a method where we put the food items in the boiling water for few seconds or few minutes and immediately take it out. By this process, we inactivate the enzymes that is present inside the food item which may decompose the food. Another method is by preventing the purely chemical reactions and by prevention of the oxidation by means of antioxidant. So basically, the self-decomposition of the food is being prevented. And the third most important principle of preservation is prevention of damage that is caused by insects, animals, or by any mechanical means. So, taking these principles of preservation, we have designed different methods of preservation. Now, preservation methods can be physical or it can be chemical. Today, we are going to learn few of the physical methods of preservation. When we talk about the physical methods of preservation, the most important thing that comes is asepsis, that is keeping out the microorganisms from the foodstuff. When we want to keep out the microorganism for the foodstuff, that means there is presence of some microorganism in the food. This is referred to as bioburden. Bio means biological entity and burden means load. That means each food has got presence of certain biological load. Now the kind and the number of this microorganism, which is referred to as the biological load or bioburden, is very important. When I talk about kinds, that means I am referring to the different types. The types of microorganism that is present inside the food may be a desirable microorganism because it can bring about a good production process, or it can be an undesirable microorganism, as well as it could be a pathogenic microorganism which is not a desirable property of the food. Therefore, when we talk about kinds, we want to maintain the number also, because if the number of the desirable microorganism is much more than the number of the pathogenic or undesirable microorganism, the preservation of the food taking place is much better. The second most important physical method is filtration. By filtration, we can clarify a liquid food. Also, by filtration, we can get rid of the microorganisms that is present inside a liquid food. By filtration, mainly we get rid of the microbes which is present in a liquid food, and this is a very important technique in the industries of wine, beer, fruit juice, and also in the dairy industry. Now, in the case of filtration, what happens is we have different types of filters. The modern filters that we use are the filters made of cellulose acetate, polycarbonate, and a variety of plastic materials like nylon, Teflon, etc. Although there are many filters which are made of sintered glass, diatoms, then unglazed porcelain, and also membrane pads. We have bacteria-proof filters. That means the bacteria-proof membrane filters which can hold the bacteria and the liquid flows out. Here you can see the picture that the typical juice is being poured inside the membrane filter. The membrane filter is present over there which absorbs all the bacteria that is present. Now this bacterial adhesion takes place in the membrane because of the pore size. The membrane can have different pore size. It can be coarse, say 8 micrometer, or it can be ultra fine, say about 0.02 microns or micrometer, which holds the bacteria. As a result, the juice which is coming out after passing through the membrane filter is totally free of microbes, and therefore we call it a sterile filtrate or a sterile juice. And therefore we get the different type of fruit juices, beer, wine, and also the water. Another most important physical method which is used for the preservation of the food item is centrifugation. Now centrifugation is a process where the sedimentation is taking place under centrifugation force. Centrifugation also helps in the clarification of a particular liquid food items. So by centrifugation, the settling down of the microbes along with the particulate matter that is present in the food takes place. Centrifugation is done in the industry in order to remove the bacteria, and therefore this is referred to as the bactofugation, that is centrifugation which removes the bacteria. And this can be done in many fruit juice industries where the storage can be increased to 22 days at 5 degrees Celsius. This is mainly done in the case of apple juice or sometimes in the milk industry where you have different milk products. So this is an apparatus which is called the bactofugation apparatus, where with the help of this the clarification of the liquid as well as the removal of the microorganisms takes place. Now the last method we are going to discuss today is drying. Now the process of drying means reducing the moisture content or the water from the food. There are different methods of the drying, and this results in production of different food items. The most important method of drying is the solar or sun drying. Now drying basically reduces the moisture content, and whenever the moisture content is not there, the microbes or the undesirable microbes are not able to grow and therefore they die. In solar drying, we are able to preserve the marine fish that is coming out or the normal fish or the flesh items under the sun. Drying by mechanical dryers is also done. Now whether it's a solar drying or drying by mechanical dryers is taking place, we are basically giving the food item a certain amount of heat. Okay, now in the case of spray drying, we have a chamber. Here you can see in this picture, this chamber has got a flow of hot air. Now hot air is flowing inside the chamber, and from the top of this chamber, the liquid, say for example, the milk is being introduced. The liquid food is being introduced, and when this liquid food comes in contact with the hot air, it gets evaporated and immediately it is changed to the powdered portion, or it gets converted to a solid state. And this solid state is being collected, and therefore the liquid milk gets converted to the solid, or we say the powdered milk, and in this way by the method of spray drying we can have the production of the milk powder. In the case of drum drying, we do not have any sprayer, rather we have large drums in the industry, and these drums are heated up because they have coils inside. Now on the surface of the drum, the liquid food item is being poured. And when the liquid food item is being poured on the drum is rotating continuously, and this liquid gets immediately converted to a solid. Again, you have production of the powder. In this way, we have production of the potato starch, and also many powdered milk is produced with the help of the drum drying process. So whether it's a drum drying process or it is a spray drying process, we are basically subjecting the heat to the foodstuff. As a result, we have the production of the powder form. A very good application of the drying is in the production of the coconut powder and the coconut milk powder. Here you can see that we take the fresh coconut and we separate out the white meat and the coconut water. The coconut water goes into some other industry for the production of vinegar or some other chemical compounds. If we take the white cooked meat and put it inside the extruder, we have production of coconut milk and the low-fat desiccated coconut. This low-fat desiccated coconut undergoes the process of drying and we have production of the desiccated coconut powder. On the other hand, the coconut milk, if it is blended and then subjected to the process of drying, we have production of the coconut milk powder, which is packed in a sterile condition. Therefore, a coconut is preserved in the form of a coconut milk powder as well as in the form of a desiccated coconut powder. Coming on to the other method of drying, which is called the freeze drying. That means drying can take place at extreme heated condition or it can take place at extreme low condition, and that is why it is referred to as a freeze drying. In the freeze drying method, the cooked food or the fresh food can be placed in a vacuum chamber where excessive low temperature is subjected, say about -50 to -60 degrees Celsius. Now, at extreme low temperature, about 90% of the moisture content of the food is being taken out. And when this food is being subjected to this extreme low temperature, after some time, it loses the moisture content, and after that, the food is sealed in a moisture and oxygen-proof packaging material. And when this food item is taken out and can be consumed, it comes into the normal temperature. So, this is referred to as a freeze drying process. Whatever the drying process is, it has to follow certain factors. For example, the temperature that is employed, the relative humidity of the air, the velocity of the air, and the time of drying. All this determines that which type of drying process is required for a particular food item. So, all these physical methods that we have studied today shows that the food can be preserved for a much longer time depending upon the type of food it is, whether it is perishable, non-perishable or semi-perishable food, and how much moisture content it retains and what is the bioburden of that food item.